Given the audience, I've tried to make this as educational and as informal as possible. So if you have any questions, feel free to stop me and I'd be happy to discuss. Maybe Nicole, if you could monitor the chat and just let me know. I'd be happy to take questions throughout, or I'm going to leave time at the end as well to answer any questions that anybody might have.
Okay.
So I assume that most of you are familiar with MRI, which is used routinely as a clinical tool for diseases that affect the brain, as shown here in this individual with multiple sclerosis. And from these images, we can see that MRI is an incredibly versatile and sensitive technology that gives doctors insight into the disease burden of a patient, which can be seen here as these bright and dark lesions and these arrows shown here. And in hospitals across the country, like Barrow, where I am right now, this imaging is used at really every stage of patient management across multiple clinical departments, including neurosurgery, radiation oncology, and neurology. And because the idea is that you can only really treat what you can see, these improvements in imaging technology have the potential to have a significant impact on patient care and ultimately the quality of life of those seeking treatment. And because of this, we've really seen an ever-increasing volume of MRI scans in hospitals, which can be a good thing or a bad thing, depending on how you look at it. For example, at Barrow, we perform over 130 scans each day and over 30,000 clinical scans each year, most of which are currently focusing on the brain neuroimaging, although there are numerous emerging applications that I'll talk about, so sort of taking these neuroimaging methods and applying them to the muscles and nerves that are sort of largely research methods right now that are increasingly being used in clinical practice and in clinical trials. And so as an engineer by training, the goal of my research in other labs is to develop these new methods and then move them from the lab into the clinic to, for example, give doctors and neurologists information on the progression of a disease, as well as how the body is responding to a specific treatment or therapy. And really, more specifically, researchers like myself, we're aiming to impact patient outcomes by improving the confidence, comfort, and care of patients through these new imaging technologies that offer, for example, higher resolution scans so we can see finer details, better contrast so we can see more sensitive to specific disease processes, shorter and more informative scans, so we have to perform fewer of these scans, which are not always very comfortable and can be expensive. And then ultimately by improving the accuracy, specificity, and sensitivity of our methods to the underlying disease processes. And so the way we do this, you can think of is by developing what we call imaging biomarkers, or you can think of this as building a better camera, which go beyond just making pretty pictures to create measures that report on the underlying biology. So again, this is like the idea of building a better camera, say like on your iPhone. On the left, we see a traditional image which appears to show this dense set of bushes, and on the right is a traditional MRI image of the brain. We see here this brain tumor, this bright region on the right, but it doesn't give us any information on the underlying biology. And so what our lab and others as well seek to do is to develop new imaging technologies that allow us to see what's happening beneath this tumor or within the muscles or the nerves, in our cases, at the cellular level, similar to the way this thermal photography method here on the right allows us to see the man that was hiding behind these bushes. And just as a field, together we call this imaging biomarker development, where a biomarker is defined as a characteristic that is a measured indicator of a normal or disease process, or often more importantly, a response to a certain treatment. So is this therapy working or not in this particular individual? And these need to be sort of, we have design criteria. We want them to be quantitative. So we want to get a number, not just is it good or bad, better or worse. We want a specific number that we can track over time. It needs to be robust. So we want the same number no matter who makes the measurement, no matter where you go to get your scan, you should get the same result regardless. And it needs to be fast and easy to use, just from a practical perspective. And that ensures that you get the same number every time. And so together, this gives us information on, it can be used to diagnose. I'll talk about a little bit. It can at least aid in the diagnosis of certain neuromuscular diseases. And then it will allow us to determine how certain emerging therapies work, whether or not they actually treat the disease or not. So in the context of neuromuscular disease, we've seen really a significant increase in the number of proposed muscle and nerve imaging biomarkers over the past decade. It's been a really exciting time. So here we show two sets of images from a baseline and a follow-up. So these are images from the calf, which show changes on the top here in lean and fatty muscle content, which gives information on muscle loss and whether or not muscles are innervated by nerves, for example, and how that changes over time. On the bottom, we see images where the brightness of the muscle gives us information of how inflamed a particular muscle is. So there's certain myopathies where that's of interest. Together, there really are a number of tools available that allow researchers to measure changes in how these important features change over time, which relates to both muscle and nerve health. And then most importantly, can be tailored to the specific diseases we'll talk about later. That's really one of the strengths of MRI in particular, is its versatility and its ability to look at multiple organs, muscles, and nerves, and different even disease processes within those organs or tissues over time. And then in addition, we can, in terms of diagnostics, we can look at patterns in how the muscles are affected. So in addition to certain clinical and sometimes genetic features, this improves the ability of doctors to diagnose certain neuromuscular diseases, as certain diseases are known to affect certain muscle groups more than others. So we can look at change over time to look at disease progression and treatment response. And then we can look at patterns in muscle involvement, which aids in the diagnosis of certain diseases. So for the remainder of the time, I thought I would just give two examples of research projects that are ongoing in our labs at the Barrow Neurological Institute. The first of which is in inherited neuropathies, or Charcot-Marie Tooth diseases, or CMT, which have a prevalence of about 1 in 2,500 and affect motor and sensory function that results in these link-dependent or dying back neuropathies, where the hands and feet tend to be most affected, and then that motor and sensory impairment tends to progress proximately towards the spinal cord over time. So we can classify CMT autosomal dominant types as CMT1, which are genetic mutations that affect the myelin sheath, so they are D or dysmyelinating neuropathies, or CMT2. These are axonopathies. So these are diseases that primarily affect the axons that transmit the electrical signals from the spinal cord to and from the rest of the body. And then these are further subtyped by specific genetic origin. For example, the most common form of CMT is called CMT1A, which results from a duplication of this peripheral myelin protein 22 gene. And then the most common axonal form is called CMT2A. So why are we interested in imaging biomarkers for CMT? Well, so one of the reasons is there's actually been a lot of developments in potential treatments for the diseases, and we really lack good measurements to track disease progression and treatment response over time, which is hindering our ability to evaluate these new therapies. And here's just an example. So, and these trials failed for various reasons, but I think it's just a good example. So there was some evidence in animal models that high doses of ascorbic acid or vitamin C slowed the progression of CMT1A. So, so that was followed up with 3 large human trials where they gave high doses of vitamin C and they measured what's called the CMT neuropathy score. So this is just a clinical score. I'll talk about it in a little bit, where the doctor goes in and asks about various symptoms and gives you a bunch of scores based on those, and then uses that symptom score to track whether or not the disease is getting worse or better over time. And what this plot on the right shows is that in the red plot here is just the placebo group. So these are the people that didn't get the high dose of vitamin C over 2 years. And you can see it's flat, essentially. There's no change. And then you see the people that receive the high dose of ascorbic acid, and there's virtually no difference. So there's a couple things here. One, the fact that CMT neuropathy score was not able to detect any change over time. So if you have some therapy that were just slow or halt the disease, you wouldn't be able to detect it with this measurement. So it's not sensitive enough to change over time. So even if, and I think more recently there's evidence that high dose of ascorbic acid probably isn't effective, but once we do have effective therapies, using the CMT neuropathy score as an outcome measure is not sensitive enough to detect this treatment response. So we need better measurements. And this just breaks down that CMT neuropathy score. The reason is that a lot of these subscores, these sensory symptoms that are measured from the doctor, are subjective. There's certain elements where the doctor will go in and measure vibration and pinprick sensibility that are, because of the link-dependent effect of this disease, even in mildly affected individuals, they can have low function scores on some on these measurements, and these result in what are called floor or ceiling effects, where they can't change anymore over time, even in a mildly affected person. And then some of the measurements aren't even available. So electrophysiology that's done sometimes is not available oftentimes in these patients, again, because of this link-dependent effect. So just to summarize what's currently available, so some of you may be familiar with some of these techniques. So one of the ways to evaluate these neuropathies is EMG, or nerve conduction studies. So that's where the doctor goes in and puts a needle under the muscle, shocks the nerve, and then measures how long it takes for that electrical signal to move down the nerve, as well as the size of how much of that propagates down the nerve. And that gives the doctor information on the health of the myelin and the axons within the nerves themselves. But unfortunately, this is often unavailable in these link-dependent neuropathies and results in non-responsive measurements. And it's also quite painful to do repeatedly. If anybody's had that done, can attest to that. You can biopsy nerve and muscle, but of course that's difficult to do over time because it's invasive. You're removing tissue. These neuropathy scores I talked about previously are subjective, and they don't give us information on all of the nerves. They tend to focus on the nerves that are near the hands and feet. It would be nice to be able to look at the entire peripheral nervous system, which imaging allows us to do. And because I talked about earlier, imaging, is quantitative. It gives us a number. It's objective. It's suited for imaging throughout the body. And so we've been developing these MRI techniques to overcome that issue I talked about in those previous vitamin C trials as potential biomarkers of disease progression and treatment response. And one of the methods we focused on is called magnetization transfer MRI. So on the blue here, we see these are nerves under a microscope. On the left, we see a normal nerve, and these dark blue circles are the myelin sheath around the nerves, and you see a bunch of densely packed circles with thick blue rings around them. That's a healthy nerve. On the right, we see a damaged nerve. So we see the rings are not as thick and it's not as dark, and there's not as many of them. So there's fewer axons and there's less myelin. So the nerve health has been affected. And our measurement, MTR, is sensitive to how much myelin, how much dark blue there is in those slides. And so that's the measurement we're going to use as a potential biomarker, because we know these diseases affect primarily the myelin sheath and the axons that the myelin sheath surrounds. And so what we've done is we've recruited 21 CMT1A, or 21 controls, and 15 CMT patients. Over 10 of those were CMT1A, 3 were CMT2A, and 3 were HMPP, which is another inherited neuropathy. We performed MRI as shown here. So the individual would lie on their back and go into the scanner feet first. We scan for about 30 minutes. We'd measure the sciatic nerve diameter, as well as the MTR of the nerve itself. And then we collected clinical data for comparison. So the Charcot-Marie Tooth neuropathy score, so that CMT-NS that I talked about earlier, which has been used as an outcome measure in previous trials, as well as these nerve conduction studies I talked about previously. And so here are just some representative images taken from the thigh. On the top are images at the mid-thigh, and on the bottom are images taken just above the knee. And then we're moving from left to right. We have a control subject. And then on the moving right, an individual with CMT1A, HMPP, and CMT2A. And of particular note, if we look at the CMT1A patient here, you see these arrows indicate the nerve, these red highlighted areas that are also indicated with these white arrows. You can see there's an enlargement actually of the nerve, which is particularly unique to CMT1A relative to any of these other cohorts actually. And then also on the far right, if you look at the CMT2A at the muscle, you see that there's quite a bit of loss of muscle at the bottom of this muscle group in the CMT2A patient, which is consistent with the idea that as muscles lose their innervation from peripheral nerves, those muscles start to shrink over time. And we actually can quantify this. So this box plot in the upper left shows in blue here that the nerve size is actually uniquely enlarged in CMT1A patients versus all these other different subtypes of CMT. And if we look at the top right panel here, we see that, interestingly, this nerve diameter is actually correlated with disability. So the larger the nerve, the more disability in this particular patient. And it's also correlated with our electrophysiology, our nerve conduction studies. So the larger the nerve, the less myelin essentially that we see within those nerves as well. So this is telling us something about the function of the patient, so we could track that over time, and it's telling us something about the pathology, about the amount of myelin in the nerve as well. So this is something that we're looking at over time to see if this tracks with disease progression and can be used as a biomarker of treatment response in future trials. And it's interesting, we see this not just so the sciatic nerve is, the longest nerve in the body. It's the nerve that goes down the thigh, but we see this also in the nerves of the shoulder here. This is showing images of the brachial plexus, where we see enlargement of the nerves really throughout the peripheral nervous system, which may give us some diagnostic insight. I mean, CMT is made diagnostically, as the diagnosis is made with a genetic test, but there are instances where imaging can aid in guiding us to the right type of test and can be used to differentiate between other types of neuropathies, for example. And then going back to this MTR imaging I talked about, so this myelin imaging. These are two maps of that myelin-specific technique I talked about. On the left, we see a map from a thigh and a control, and in that zoomed in set, we see the sciatic nerve, and we see these yellow values, which are indicative of high amount of myelin. On the right, we see an individual with CMT1A, again, zoomed in set, we see the nerve itself, and we can see these green hues, which are indicative of a lower amount of myelin, which is consistent with what I talked about previously. And again, we also see this enlargement of the nerve, which is just specific to CMT1A. And like I talked about in these nerve diameter estimates, we see that there are differences across different types of inherited neuropathies for MTR. So controls have the highest MTRs you would expect, CMT1A or lower, and then these axonopathies, so CMT2A, which tend to be more severe, have the lowest amount of myelin and the lowest amount of MTR. So this is telling us something that might be indicative of disease status across all these various different types of inherited neuropathies, whereas nerve diameter might be something that's just specific to CMT1A. And in fact, if we look at correlations between, so the y-axis here is this MTR, this myelin measurement from MRI, and the x-axis is this CMT neuropathy score. So this is this clinical score of disability. And what we see is that as disability increases, MTR decreases, and this is across all different types of CMT. So it's not specific to a certain subtype. So this might be a nice general biomarker for all different types of inherited neuropathies. There's also a lot of work by other groups and ourselves in looking at the muscles. So these neuropathies affect the nerves directly, but downstream the muscles are affected. So as the muscles lose their innervation from the nerves, they start to shrink. And as the muscles shrink, the area that was occupied by the muscle is eventually replaced by fat. And so we can use a technique called Dixon or fat water imaging to quantify how much fat is in the muscle. And this is what these researchers have done here. So on the top left is a control subject, and we see basically the muscle is dark everywhere, which means there's almost no fat in the muscle, which is to be expected. And on the bottom, we see a patient with IBM, an individual with IBM, and you can see that the muscles are much brighter, so there's much more fat, which is indicative of, again, these muscles losing innervation and shrinking, and then eventually being replaced by fat. And so we can monitor how muscle fat changes over time, and that can be used as a biomarker of disease progression and treatment response. But it turns out that in many cases, and it really depends on the application, but direct measurements of nerve, at least for neuropathies where the disease originates from the nerve itself, might be better, and that's what we've shown here. So in the y-axis here is MTR, so this myelin measure, and the x-axis is disability, and we see that the relationship between the two is much stronger in the nerve than it is in the muscle, at least for peripheral neuropathies. So that might be a better biomarker. But the nice thing about imaging is that it's incredibly versatile, and we can look at both simultaneously. So we can look at both muscle and nerve health and how the two are related and affecting one another, and get a more complete picture of the current disease status and how things are changing over time. And this really is, probably disease specific. If we were looking at myopathies, for example, then it may be better to look just at the muscles themselves. And then one of the things that we're doing right now is looking at, okay, so we've shown that these measurements relate to disability and the amount of myelin, but how sensitive are they to change over time? And here's just an example. So we scanned a patient in blue at time point 1 and in red at time point 2, and you can see that overall the MTR values dropped pretty dramatically in this particular patient over time, which is indicating that we are able to detect a progression of the disease over time that wasn't detectable using these current clinical scores that are available. So we're actually recruiting more patients right now. I'm working with researchers at Wayne State to do a longitudinal study to evaluate these methods. So second example I wanted to discuss this morning was ALS, which is a motor neuron disease, a rapidly progressing motor neuron disease. And the evaluation of emerging therapies right now is really hindered by a lack, again, just like CMT, of objective and sensitive measures that track with disease progression. As shown here in this image on the right, so this is just your standard clinical whole body muscle image. We know that muscles are brighter on MRI in ALS individuals compared to controls. However, this bright signal is, although it's associated with both disability and clinical weakness, it doesn't really give us any specific information on the disease itself and how it's progressing and how it's responding to a given treatment. So that has led researchers like ourselves at the Barrow Neurological Institute to develop new methods that give us insight into what's going on, sort of like that better camera, at the molecular or cellular level. And the method that Dr. Quarles and his group at the Barrow Neurological Institute have developed is this idea of, they call it MR cytography. And the idea is that MRI images water and its magnetic properties, but you can also give dyes or contrast agents. And these contrast agents will go into the blood vessels and they'll leak out of these blood vessels into the muscle tissue and surround all the muscle cells. And one of the properties of our MRI images, called T2 star, depends heavily on the spatial distribution of this dye or this agent, which is determined by the local cellular structure. So how big are the muscle cells? How are they packed? How many of them are there, et cetera. So we can make a measurement that's sensitive to the local structure of muscle. And it turns out, so this is on the left, we see these are muscle images under a microscope of a wild type mouse and a mouse that has a genetic mutation that's a model of ALS on the bottom. And you can see that the size and the packing and the shape of the cells is very different in these two muscles. So the distribution, like I talked about on the last slide, of the dye, the contrast agent, is going to be very different in those two instances. And it's going to give us a very different signal property, which we can measure with MRI. And in fact, that's what they've shown. So the measurement they get from that is called trait. And what they've shown is that this, on the left here, we see trait measurements in these white box plots measured at 9 weeks, 12 weeks, and 15 weeks in a wild type, just a normal healthy mouse. And on the right, in these gray, we see similar values at 9, 12, and 15 weeks in this ALS model mouse. And we see stability in the healthy trait values, but a steady decrease in the ALS mouse, which is telling us that this might be measuring something specific to the way the disease is progressing over time. And then so more recently, they've applied this to individuals with ALS. So they've recruited 10 individuals or 12 individuals with ALS and 10 healthy controls. They've also collected these clinical measurements as ALS, FRS, and HDD, sort of standard clinical scores similar to the CMT&S I talked about previously, but tailored obviously for ALS. And they looked at different muscle groups. So is this the same in all muscle groups, or is there a pattern that's different across different muscle groups like we've seen previously? And this just gives you an idea of sort of the versatility of imaging. So, in a 30-minute exam, we can bring a person in, get anatomical images of the calf as shown here, which give us some information on the pattern of muscle involvement. We can do this fat water imaging, which measures muscle atrophy and fat replacement. We can get some pre-contrast imaging and then give this dye and then look and see how the signal changes over time as this dye is taken up and distributed around these different muscle cells, as I talked about, from which we can measure that trait measurement which tracks with disease progression in these mouse models, as I shown previously. And then from all this, within a 30-minute scan, we get a wealth of information. So we get your standard T1 weighted images, but also these trait measurements, again, which is the biomarker that we're proposing, along with fat fraction, which measures muscle atrophy and fat replacement, as well as T2 maps, which measure muscle edema. So that is once again the power of MRI, is the ability to look at multiple potential disease process simultaneously within the same individual within a relatively short amount of time. And so what they found was similar to what I showed previously, is that there's a heterogeneity. So the trait values differ quite a bit across different muscle groups. And then most importantly, it was consistently lower in the ALS patients compared to healthy controls. So it might be a potential biomarker similar to what we saw in the mouse models. And then if we compared this trait measurement to more traditional muscle-based fat water imaging, we see that trait was almost 4 times more sensitive, or the differences we saw were about 4 times larger than for muscle-based, or for these fat water imaging. So it might be a more sensitive and specific biomarker of disease progression. And so they're looking into this further moving forward. So as I said, this MR cytography method has the potential of being a robust, non-invasive, quantitative biomarker to evaluate ALS-induced changes to muscle structure, which is known to be affected in ALS. And they are looking at imaging more patients and then comparing it to other non-imaging reference standards and looking at how robust the measurement is. So do we get the same value if we scan it over multiple time points in the same individual? And then looking at the change over time. So I showed some results in animal models with change over time, but they're doing the same studies in patients to see if they can detect a change over a 6-month period. So where do I see neuromuscular MRI heading? I think we're really starting to see many of the methods I discussed move. They've lived mostly in the lab into clinical trials, increasingly to evaluate these new exciting therapies, and increasingly into clinical practice even. That will allow doctors to see the unseen at higher resolution and contrast than is currently possible. And most importantly, with amplified sensitivity to the underlying disease or biology. Furthermore, I think research will continue to push these methods out of the lab into the clinic so that I hope all doctors and patients have access to these tools, so these aren't just boutique tools. As an example, we just recently applied for a grant through the NIH, which aims to develop these methods for all major MRI vendors across multiple sites, including Wayne State, which is the home of the world's first CMT clinic. And if successful, these methods will improve our confidence in clinical decision making and our ability to diagnose these diseases better, improve patient comfort during scanning, and then ultimately improve the care of these patients and individuals by evaluating disease progression, and more importantly, responses to emerging therapies. So in closing, I just wanted to thank all the people in my lab and our collaborators at the Barrow Neurological Institute, Wayne State, my previous institute at Vanderbilt back in Nashville. We've had support from commercial partners, Philips, our scanner, commercial partner, and the government agencies that help us perform this research, the NIH and the Department of Defense. And most importantly, I just want to thank all of you for the opportunity to share an overview of this exciting field and give some examples of our research and research of others. And I, of course, would be happy to take any questions that you might have at this time.
Thank you, Dr. George. I was curious, you had mentioned that the longitudinal study, you were recruiting patients for that study. How would someone find out more information about that?
They could contact me, and then I could forward you to our research coordinator. That would be the easiest
Okay.
um, method at this point.
Okay.
Um, yeah.
It's definitely interesting. I can definitely see the benefits of this over a nerve conduction study and a biopsy. How
Yeah,
do you,
absolutely.
how do you see this moving in the future with neurologists across the country? I mean, I think that it's going to probably expand and become more widely used, like you said, for the future. What are your thoughts on the future with other neurologists using this technology?
I do think that it depends on the specific um, disease, but so I think the most immediate impact is in clinical trials. So, like, we don't have good ways to know whether or not a drug is working well or not.
Mm-hmm.
Especially in a slowly progressing disease, it's very difficult because you're looking at very subtle changes over time.
Mm-hmm.
So these methods in the next decade will, I think, will definitely be used as measurements to look at the progression of the disease and then how that progression is changed once you give a certain drug or treatment.
Mm-hmm.
That's the biggest challenge right now we're facing in these clinical trials is we don't have a good way to know whether or not the drug is working or not.
Mm-hmm.
Uh, and then I think over time, as these are established, then they start to filter into your standard clinical practice. So, instead of maybe getting a nerve conduction study, can we do this nerve MTR instead? Because you said it's, for one, nerve conduction studies aren't available in all individuals.
Mm-hmm.
And then secondly, they're quite uncomfortable and painful. So being able to do sort of a quote unquote virtual nerve conduction study with imaging or something like that would be maybe a little further off in the distance, but it's
Mm-hmm.
one of our goals.
Well, that's great. And that's one of the reasons why I wanted to have you on today is talk about this. I think it's definitely new information for a lot of people out there. And hopefully this is something that we can see that will happen in the future. Thank you very much for being with us today.
All right. Thank you.
Have a good rest of your day.
All right. Bye-bye.